In this class, we are going to study about nitrification and denitrification process. The contents of the lecture include forms of nitrogen in wastewater, why the forms of nitrogen treated, then the detailed process about nitrogen cycle that include nitrogen fixation, assimilation, ammonification, nitrification and denitrification. In overall nitrogen cycle, nitrification and denitrification process are most important. Nitrification is nothing but the biological conversion of ammonium to nitrate nitrogen. Then denitrification is the biological reduction of nitrate to nitrogen gas. That means nitrate obtained from nitrification process is reduction to nitrogen gas. It is called denitrification. Before studying about the nitrogen and denitrification process, we should know the forms of nitrogen which are available in wastewater. In this, number 1, total inorganic nitrogen. The total inorganic nitrogen is the sum of ammonia plus nitrate plus nitrate. These are the combination of ammonia, nitrate and nitrate which are present in the wastewater. Number 2, total Gegendal nitrogen. It is simply called as TKN. It is nothing but the sum of organic nitrogen plus ammonia. Number 3, ammonia. When the pH of the wastewater is acidic or neutral, the majority of the nitrogen is ammonium. Generally, when pH increases over 8 in the wastewater, the nitrogen is mostly found in the form of ammonia. Normally, a municipal wastewater treatment plant with an effluent containing more than 2 or 3 mg per liter of ammonia. Fourth one, organic nitrogen. It is a small fraction, typically 1 or 2 milligrams per liter. Normally, a municipal wastewater plant containing less than 3 mg per liter of organic nitrogen. Fifth one, nitrate. A municipal wastewater effluent is containing less than 3 mg per liter of nitrate and its maximum concentration may reach up to 20 mg per liter. Sixth one, nitrite. Municipal wastewater effluent generally contain less than 1 mg per liter of nitrate. It is lesser than the concentration of nitrate. And the seventh one is nitrogen gas. We all know our atmosphere containing 78% of nitrogen and 21% of oxygen, and the remaining 1% is argon and other inert material. Let us know why it is necessary to treat the forms of nitrogen. The reasons are: it improves the receiving stream quality in wastewater plant, increases chlorination efficiency, minimizes pH changes in the plant, and increases the suitability of reuse. Next, it prevents ammonia toxicity, protect groundwater from nitrate contamination, to control the growth of algae, to prevent dissolved oxygen depletion and some odor problem. Next, to prevent the imbalance of natural ecological system and increase of eutrophication. And last one is increase the risk of human health such as nitrogen concentration in drinking water. Next, let us see the detailed process of nitrogen cycle. Normally, the nitrogen cycle is occurred by the 5 process. One is nitrogen fixation, second one is assimilation, third one ammonification, and the most important are nitrification and denitrification. Nitrogen fixation is the fixation of atmospheric nitrogen into organic nitrogen. This process is carried out by many of the microorganisms, generally called as nitrogen fixing microorganism or nitrogen fixer. The nitrogen fixation is occurs by 3 ways. One is lightening strike, second one is fixation by free living bacteria, and fixation by symbiotic bacteria. Among these 3, most fixation is done by free living and symbiotic bacteria. These bacteria containing nitrogenase enzyme that combines gaseous nitrogen with hydrogen to produce ammonia. Then the ammonia is converted into other organic compound by the action of this nitrogen fixing bacteria. Good example for free living nitrogen bacteria is Azotobacter, and symbiotic nitrogen fixing bacteria is Rhizobium. Next process is assimilation. It is nothing but the absorption of nitrate or nitrate ions by the plant root. Generally, the plant cannot assimilate ammonium ions directly, so at first nitrate is absorbed by the root, then it is reduced to nitrate ions, and then with the help of ammonium ions, the plant converts the nitrate ions into amino acid, nucleic acid and chlorophyll. This process is carried out by animals, fungi and many heterotrophic organisms. Then nitrification. It is the biological oxidation of ammonia or ammonium to nitrite followed by the oxidation of nitrate to nitrate. You can clearly see with the image. In this image, the ammonium react with oxygen, then it form nitrite. Again, the nitrate is oxidized into nitrate. I already told different forms of nitrogen is present in the environment, which include ammonium, nitrite, nitrate, nitric oxide, nitrous oxide, inorganic nitrogen gas and organic nitrogen. The nitrification is a microbial process by which a reduced nitrogen compound are sequentially oxidized into nitrate and nitrite. This process, that means the conversion of ammonium to nitrate, is performed by many soil living bacteria and other nitrifying bacteria. The nitrification process occurred by two steps. In the first step, the ammonia is converted into nitrate with the help of oxidizing bacteria which are oxidize the ammonia. The best example is Nitrosomonas, which convert ammonia to nitrate. In the next step, the nitrate is converted into nitrate. Again, with the help of oxidizing bacteria, the nitrate is converted into nitrate. Nitrobacter is a good example of oxidizing bacteria which convert nitrate to nitrate. The last process is denitrification. In this process, the reduction of nitrates back to the largely inert nitrogen gas, which completing the nitrogen cycle. Generally, this process is performed in anaerobic condition. That means the process is mainly carried out by anaerobic bacteria. The denitrification occurs when the oxygen levels are depleted and nitrate becomes the primary oxygen source for microorganism. The denitrification occurs when oxygen levels are depleted and nitrate becomes the primary oxygen source for microorganism. Generally, the process is performed under anoxic condition. That means the dissolved oxygen concentration is less than 0.5 mg per liter. It is ideally less than 0.2 mg per liter. In this process, when bacteria break nitrate to gain oxygen, the nitrate is reduced to nitrous oxide and then nitrogen gas. The best formula describing the nitrification reaction is 6NO3 plus 5CH3OH will give 3N2 plus 5CO2 plus 7H2O plus 6OH. Denitrification is an alkalinity producing process. The optimum pH values for denitrification is ranging from pH 7 to 8.5. In this process, approximately 3 to 3.6 pounds of alkalinity is produced per pound of nitrate. Then the denitrifying bacteria can use either dissolved oxygen or nitrate as a source of oxygen for their metabolism. And the most important thing is the denitrification process only occur under anaerobic condition. Another important aspect of denitrification is the requirement for carbon. That is, the presence of sufficient organic matter to drive the denitrification reaction. Normally, the organic compound may in the form of raw wastewater or supplemental carbon, and the following condition which are mainly affect the efficiency of denitrification process, which include nitrate concentration, anoxic condition, presence of organic compound, pH, temperature, alkalinity and the effect of trace metal. These are the factors which commonly influence the process of denitrification. The most important features of denitrifying bacteria is it is generally less sensitive to toxic chemicals compared to nitrification bacteria, and also they can recover from toxic shock in a shorter time. Then mainly the temperature affect the growth rate of denitrifying organism. Normally, the denitrification process can occur between 5 and 30 degrees Celsius, and this rate increases with temperature based on the type of organic source which are present in the sewage system. The highest growth rate can be found when using methanol or acetic acid. and the lowest growth rate, which are found when recycling on endogenous carbon sources at low temperature. These are the major factors which affect the process of denitrification. Next, we should know the common microorganism which carried out denitrification process. The good examples are Nitrobacter, Nitrosomonas, Paracoccus, Achromobacter, Thiobacillus, Micrococcus, and some species of Serratia, Pseudomonas, and Achromobacter. Apart from this, Pseudomonas aeruginosa in anaerobic condition can also reduce the amount of fixed nitrogen. That's all about the nitrogen cycle. Next, we will discuss about the anaerobic digestion of wastewater by methanogenesis. The contents of the study include types of methanogenic anaerobic digestion, microbiology of methane digestion, and various process parameters that include temperature, pH, and nutrient supply. Wastewater is nothing but used water. It may be from household, agriculture, and many industrial purpose. The wastewater containing high concentration of many degradable organic material. When it's processed with various bioreactor, it will produce a valuable bioenergy or many biochemicals. Among various bioenergy process, the methanogenesis is one of the important digestion process which takes place in the wastewater treatment. Methanogenesis is a biological production of methane from either carbon dioxide and nitrogen or from any methylated organic compound. This process is strictly anaerobic process. The bacteria which are involved in the methanogenic process are called as methanogenic bacteria. They are also called as methanogens. Most of the methanogens are belongs to archaeal group. That means archaebacteria. The archaebacteria can be classified as either chemolithotropic methanogen and methylotropic methanogen. The most important feature is they are extremely oxygen sensitive. It only grow in the absence of oxygen. The methanogenic bacteria are commonly found in digestive stream of herbivore, marshes or lake bottom, many mud sediment, and in man-made anaerobic digester in sewage treatment plant. I already told the methanogens are classified into two types, chemolithotropic methanogen and methylotropic methanogen. The chemolithotropic methanogen that grow only in the presence of carbon dioxide and nitrogen. But the methylotropic methanogen that grow with methyl group containing any substrate. For example, it may containing methanol, methylamine and acetate. Now we will see the methanogenic anaerobic digestion. The anaerobic digestion is more popular than compared to aerobic method which are involved in the wastewater treatment because it is more simple in construction and operation. This process required no external power. Then compared to other digester, it is very compact. They generate little biological sludge waste. Then it has high treatment efficiency compared to other bioreactor. The process has low capital and operating cost and it does not require oxygen. This is the important one. And finally, the most important thing is it produces methane. We will use methane as a fuel. Let us see how the organic material present in the wastewater is converted into methane. Generally, the process has four steps. In the first step, the fermentative bacteria hydrolyze some complex organic polymer which are present in the sewage such as protein, polysaccharide and lipid into amino acid, sugar and fatty acid. That means the complex organic material is converted into simpler form. In the second step, the simplest monomer are then fermented to intermediates containing a mix of organic alcohol and acid. The third step is called as acetogenesis. Here, the organic alcohol and acids are further oxidized into acetic acid, hydrogen and carbon dioxide with the help of hydrogen producing acetogenic bacteria. And the last final step is methanogenesis. In this process, the methanogenic bacteria convert acetate into methane and carbon dioxide. This is the final product. The methane can be used in the production of biodiesel. Let us see the methanogenesis process in detail. I already told the wastewater containing many of the organic material which may include carbohydrate, fat and proteins. In the first process, the large organic material such as the protein, fat and carbohydrates are converted into simpler form. That means the carbohydrate is converted into simpler sugar, the fat converted into fatty acid and proteins converted into the simpler amino acid. The breakdown of organic material is catalyzed by many of the hydrolytic enzymes. It may be a protease or amylase or lipase, which are produced by many of the bacteria. For example, Bacteroid, Clostridium, Fusobacterium, Selenomonas and many bacillus species. In the next process, that means acidogenesis, the simplest sugar, fatty acid and amino acid are converted into carbonic acid and alcohol and also hydrogen, carbon dioxide and ammonia. In the third step, the acidogenesis. In this reaction, the carbonic acid, alcohol, hydrogen, carbon dioxide and ammonia are converted into hydrogen, acetic acid and carbon dioxide. This process is carried out by many acidogenic bacteria which convert the hydrolysis product into already told hydrogen, carbon dioxide and organic acid. Some of the example of acidogenic bacteria are Clostridium, Lactobacillus and Fusobacter. These are the genus which are commonly involved in the acidogenic process. The last step is methanogenesis. That means the production of methane and carbon dioxide. In this step, the simplest form of hydrogen and acetic acid are converted into methane. This step is carried out by many methanogenic bacteria. For example, Methanococcus, Methanosarcina, Methanosarcitae and Methanogenesis species. Next, the important factors which are involved in methanogenesis process. They are temperature, pH and nutrient. The anaerobic digestion of wastewater is facilitated by a population of microorganisms in the absence of oxygen. So, it is important to maintain certain environmental conditions in order to ensure the optimal function of microorganism. The first parameter is temperature. Normally, the microorganism convert the wastewater into methane under mesophilic and thermophilic condition. That's why the anaerobic digestion plants are generally operated at 35 degrees Celsius. Normally, the effluent usually enter the digestion system at the temperature below 18 degrees Celsius. So, more heat is required in order to carry out the methanogenesis reaction. So that in total material, 30% of the waste will be consumed in order to raise the heat. Commonly, the generation time of methanogenic bacteria is ranging from 3 days at 35 degrees Celsius and 50 days at 10 degrees Celsius. Thus, it is important for wastewater plant in order to operate at high temperature because a prolonged regeneration time lowers the biochemical activity and efficiency, which ultimately result in low methanol yield. Next parameter, pH. Normally, the acid producing bacteria grow under the range of 5 to 6, but the methanogenic bacteria grow at the optimal range of 6.7 to 7.4. The methanogenic bacteria are highly sensitive to pH, so the anaerobic wastewater treatment system will operate at a pH between 6 to 8. In this condition only, we can obtain high growth of methanogenic bacteria. Suppose the pH of the sewages get lower, the activity of the methanogenic bacteria can also get lower. The third one is nutrient. In anaerobic digestion, the growth of microorganism will require nitrogen, phosphorus and some trace elements such as magnesium, iron, nickel, potassium, sulfur, calcium, cobalt, zinc, manganese and copper. These trace elements are commonly found in the most wastewater, so we need not to supply in the seed sludge. In some cases, suppose the sewage containing some heavy metals like copper and zinc, they may affect the growth of acidogenic bacteria. That leads to the reduction of methane formation. The advantages of anaerobic digester include production of methane and carbon dioxide, which can be collected and used as a fuel. The process reduces the volatile content of the sludge. In this process, the liquid is more easily dewatered as liquid and solid from separate layer and also reduction in number of pathogens. These are the some advantages of anaerobic digester. This process also have some disadvantages. They are the process continuously need constant supervision. Next, there is a chance to formation of some explosive gases. In this time, it is not easily controllable. The methanogenesis process is a slow process. Also, the maintenance and cleaning is somewhat difficult. That's all about methanogenesis. Thank you.